All right, hello everybody. Thank you for tuning in for my presentation. In the coming 10 or 15 minutes, I'll tell you a little bit about our recent research and how we reconstruct absolute seasonality using clumped isotopes in bivalve shells. So before I start, I'd like to thank a few people. First of all, I'd like to thank the funding that made this research possible. I'm funded by the Flemish Research Council as well as the Marie Curie funding from the European Commission. And then there's a range of people here from different institutes that contributed in different ways to the work that I'll be presenting in the coming minutes. So I'd like to thank them all. I'd like to start by introducing my topic with this slide here. And starting on the left side here, you see a graph that might be familiar to most of you if you're into paleoclimatology. And that is showing that if we go further back in time, millions of years, we'll find climates like in the Eocene, for example, or the Pliocene, that were warmer than we have today. And these climates are interesting to study with an eye for the future, because we might arrive at climates that are much warmer in the future, of course, due to anthropogenic global warming. So interestingly, to have some analogs for different warming scenarios in the future, we have to go back in time and do paleoclimatology research to tune our models, make them better in predicting climate. Disadvantage of this is, of course, if you go further back in time, it gets more and more hard to reconstruct climate at a very short time scale. And that's where I want to move to the figure here on the right side of the slide. And it shows you the kind of variability that happens in climate system over time. This is a logarithmic scale on the bottom here. So these are years, these are millions of years. And what you see is that a lot of this very important variability in climate happens at a very short time scale. We're talking about seasons, tides, days, that kind of variability. And it becomes progressively harder to reconstruct this type of climate variability when we go back in time. So we need good archives to do this, because this type of short-term variability is very important for us because it happens at a human timescale. So I would argue that one of the best archives to do high-resolution climate reconstruction, and specifically seasonality reconstruction, are fossil bivalve shells. As most of you might be familiar, bivalve shells grow incrementally. In this picture here of this fossil oyster shows this very nicely. They grow over the seasons, and they record these nice little increments in their shell. And while they grow, they record their environmental conditions in the chemistry of their shell. And we can do all sorts of fun high-resolution analysis on the shells. And because the shells are made of carbonate, they grow a bit like tree rings, you could say. But because they're made of carbonate, they preserve much better than, for example, a tree ring would. So we can use fossil bivalve shells for, in this case, for example, millions of years in the past, more like 67 million years, but I'll show you some older examples also to reconstruct a climate at a very high resolution and very far back in time. It makes it ideal for this type of work. In my project called Unbiased, I combine actually several methods, several avenues of research to make this archive of bivalve shells better and to improve the way we can reconstruct this type of high resolution variability. I will mostly be talking about the clumped isotope aspect of my work here, and also a little bit about the statistical routines that we use to get the most information out of these records from bivalve shells. So to start a little bit of introduction about clumped isotopes. As some of you might be familiar with paleoclimatology, we use isotope ratios in carbonates, and most predominantly the oxygen isotope ratio that you see here, as a measure for temperature, as a paleothermometer. And in oxygen isotope ratio, we look normally at the ratio between oxygen 18 and oxygen 16, which gives us some information about temperature. The advantage of this method is that we need very little material, less than 100 micrograms to measure oxygen isotopes because they're quite abundant. But the disadvantage is that to be able to reconstruct temperature from these measurements, we need to know something about the oxygen isotope composition of the seawater in the past, and it becomes also progressively harder if we go back to deep time settings. And that's where the clumped isotope method comes in. So in the clumped isotope method, we measure the abundance of certain isotopologues, so molecules of carbonate that contain both a rare carbon and a rare oxygen isotope. And this measurement is independent from this seawater delta ATO value. It just measures only the temperature. The disadvantage of this method is that you can already see that these isotopologues with two rare isotopes are even more rare than the rare oxygen isotopes. So we require more material. So that's why in my research I work on ways to get around this problem of requiring more material so we can get high resolution records from this clumped isotope technique. So there's several ways in bivalve shells to do this. If we are interested in the seasonal cycle, we can measure clumped isotopes in very small samples in our lab in Utrecht. But the error bar on individual measurement is very big, so we need to combine different measurements from the different seasons to get a good error bar on the summer and winter temperature and still be able to reconstruct seasonality independently from this oxygen isotope composition of the seawater from a bivalve shell. And there are several ways to do this. So this is the normal oxygen isotope method, and we can kind of smooth our record or bin them monthly if we have a good age model, which we often have in bivalve shells. And I would argue we tested different of these variation of these methods on a range of virtual data sets and also natural ones. And you can find details about this testing in our paper that's going to come out in a few weeks. And actually, if we do this, we discover that there's a way in which we can do this most efficiently, this combining of measurements from the summer and winter. And it works as follows. We basically we measure a profile for the bivalve shell, and we combine measurements from the summers and from the winters together and find the optimal compromise between having a very high peak summer value and still combining enough measurements to get a low error bar. So there's a way statistically with the moving window t-test to find out this method. I can explain more about that in the questions if you have them. So to apply this method, our first study where we applied this is on this very nice site from the Kristianstad Basin in southern Sweden. So this is a late Cretaceous site that's 78 million years old, and it contains very excellently preserved bivalve shells because this Baltic shield here in the Scandinavia has not been subject to a lot of tectonics since this time. So these shells have not been buried much, and they're very well preserved, which is very important for clumped isotope measurements because it's very sensitive to diagenesis. So we tried our method on several different specimens, oysters, but also Rudis, which is the most northerly occurrence of Rudis bivalve shells that we know from the Cretaceous, so that's very interesting as well. And if we compare the results, we see that our results from different species line up quite nicely, and we get very nice monthly temperature reconstructions using this clumped isotope method. But even maybe more interestingly, we also find that there's a very big effect on the delta 18 of the seawater, which we can of course back calculate. Once we have the temperatures independent from the clumped and we have the delta 18 values, we can calculate what this value is. And we see that there's quite significant difference over the seasons, which is something that should concern people if they're using normal oxygen isotope composition for temperature estimates. The other conclusion here from our work is that we compared our reconstructions in a study that's just been published a week ago. We compared our reconstructions with models from this time, climate models, and we showed that our reconstructions on the seasonal level actually match very well with the model, and they actually show somewhat warmer and more seasonal temperatures than previous reconstructions from the same latitude, which is interesting. So it means that we have quite extreme seasons in the Cretaceous when we have this greenhouse state, which has implications, of course, for future climate and for improving our models, which is something a result that we were very happy with. Tell you a little bit about the future. We're currently working on this method and taking it sort of a step back, more or less closer to home in literal sense. The next project we're working on now is the Pliocene warm period, which is a very interesting period, about 3 million years ago, in terms of climate and greenhouse climate and constructions for the future, because Pliocene warm period had about the same level of CO2 that we have today. So it's a nice analog for if we would limit our CO2 emissions to the level that we are on today and let it run to equilibrium, we would end up at this Pliocene climate, which is approximately 3 degrees warmer. We also have a very good model basis for the Pliocene warm period. There's lots of models and model intercomparison studies from this period. And we have very excellent aerogenitic preserved shells from Belgium and the Netherlands that we're currently working on with some very nice, excellent students of mine. And we'll be applying the clumped isotope seasonality method to characterize the seasonality during this Pliocene warm period to have an idea about the seasonality that we might be heading towards in the future and compare them with models. So these shells originate mostly from the Antwerp Harbor region in Belgium, and we have some shells from the Pliocene warm period, but also some from before and after. And I'll show you here some initial results just for the oxygen isotope composition, because the clumped isotope are still a work in progress. We're actually measuring them this week. And you see already that all these species of bivalves from this Pliocene warm period show these nice seasonal patterns, and we can now target the summer and winter parts of these records for clumped isotope analysis and get some very nice estimates of seasonality in the Pliocene warm period. So I hope to be able to tell you a little bit more about the results of this study in a later time. And in the meantime, of course, I hope you enjoyed my presentation, and looking forward to see you during the Q&A. If you have any questions, I'll be happy to answer them there. Thank you very much for your attention, and have a nice conference.